The reason why it's important to study plants in space is that plants are absolutely the things that will allow us to travel past the restrictions of a picnic basket, right? We're not going to be able to explore our solar system, explore our world past low Earth orbit unless we can take plants with us, because that will enable us to grow our food, to recycle nutrients, recycle our air. This is what it's going to take. What Rob Ferl and I do is we engineer the plants to be tools of exploration. We've engineered them with green fluorescent protein molecules that allow us to follow how particular genes are utilized in the spaceflight environment versus the terrestrial environment. One very effective way to take a look at these engineered proteins is to be able to have a tool that looks inside the cell. We've done a lot of work by looking at the whole plant and how they grow, even with green fluorescent proteins, to get a sort of a bigger picture of how things are turned on and turned off. But being able to look inside the cells is extremely powerful. And in order to do that, we needed a microscope. We used the light microscopy module on the space station, or the LMM. What that enabled us to do is to take a look inside specifically how certain signaling molecules that are known to be important to gravity signaling on Earth, to see how that functions in the microgravity environment. We looked at two signaling pathways, or genes associated with signaling pathways. One was for auxins and one is for cytokinins. Both auxins and cytokinins are plant hormones that are important to plant growth and development. And what we found was that the auxin type signaling molecules seemed like they were pretty much unchanged, whether you were in a no gravity environment or in the terrestrial environment. You saw essentially the same patterns of distribution in root tips of auxins that you do on the Earth. However, in the other hormone, cytokinins, we saw that you did see a different pattern of the way cytokinins were distributed in the ground controls versus what you see in spaceflight. And so what this did is this enabled us to take a look at how plants physiologically adapt to microgravity, and also was kind of some insights into, well, maybe how some of these things work even in our own backyard. But what we have done with this little tiny weed, Arabidopsis Thaliana, which is the sort of the white mouse of the plant world, it's a model organism, is enabled them to tell us more than just how they grow. It's enabled us to tell us how they do the substrate of what makes them grow. And that's what this genetic engineering can enable us to do, more so than just your normal garden variety plant that you send up.